experiments, need experiments in cell biology, a video.
Bulk and single molecule assays for cytoskeletal activity. Much of our knowledge about the mechanisms that regulate the function of the cytoskeleton has come from investigating the activity of purified proteins in a test tube. A key aspect of cytoskeletal function is that individual subunits can assemble into cytoskeletal filaments, such as actin filaments or microtubules. The dynamics of this cytoskeleton assembly and disassembly are important to understand because these processes are largely responsible for the movement of cellular structures and the mechanical forces that allow entire cells and tissues to move. To understand how assembly is regulated in a cell, researchers have developed in vitro assays to measure filament assembly from purified subunits. These assays are artificial in that they remove many of the proteins that might be working with the cytoskeleton in the cell. And also they remove physical structures that could be important for organizing cytoskeletal structures, such as vesicles or the plasma membrane. However, by increasing the complexity of the reconstituted system, researchers can effectively use these assays to identify the fundamental properties that regulate the cytoskeleton and the minimal set of proteins required for in vivo behavior. Let's review the pyrene-actin assay, which monitors actin polymerization. Remember, the actin can exist in a globular form called G-actin, and this G-actin can polymerize into a filamentous form called F-actin. What do you need to assay actin polymerization? First, you need purified G-actin. Researchers often start with rabbit muscle, a tissue that is highly enriched in actin, to purify this protein. Next, you need a way to determine whether the G-actin subunits have polymerized into F-actin. To do this, you can covalently attach pyrene, a fluorescent molecule, to a cysteine in actin by performing a chemical reaction. We know that pyrene-labeled G-actin is still able to polymerize, and that polymerization greatly enhances the pyrene fluorescence. Thus, by simply measuring the relative increase in fluorescence, you can monitor the amount of polymer in a reaction because these values are proportional. In addition, by monitoring fluorescence over time, you can examine the kinetics of the assembly reaction. Thus, you have an assay to measure the transition from G-actin to F-actin and vice versa. The pyrene-actin assay has been extremely useful for understanding the kinetics of assembly and for determining how proteins can modulate the assembly-disassembly reaction. However, because you are measuring the total amount of fluorescence, not observing the pattern of fluorescence using a microscope, there are limitations to this assay. One is that it is a bulk assay. Your readout is a measure of the behavior of a population of filaments at any given time. Think about this. Could you theoretically have the same pyrene fluorescence if you had many short filaments, for example, via nucleation, or if you had a few long filaments? Yes, details such as the length of the filaments or how fast individual ends of the filament elongate are not resolved with the pyrene-actin assay. But you can still get a good idea about how a protein influences actin polymerization overall. For example, a protein that nucleates filaments will reduce the initial lag phase and result in a greater rate of fluorescence increase. Another limitation of the pyrene-actin assay is that you do not see the morphology of the filament or where an actin binding protein might interact with the filament. As Yogi Berra says, you can observe a lot by just watching. A more ideal experiment may be to directly visualize individual filaments and even the actin binding proteins that influence assembly or disassembly. So how can you visualize individual filaments? First, you will need to somehow label the actin. Instead of labeling actin with pyrene, you could label actin with a fluorophore that fluoresces in the visible range, like an Alexa fluor, making it possible to use a fluorescence microscope to visualize the actin. However, there is one challenge with this approach. You will need a technique that allows you to specifically distinguish actin polymers as they grow, without getting lost in the background fluorescence produced by labeled G-actin in solution. So to solve this problem, could you selectively illuminate the F-actin? One optical technique now widely used in many fields is called total internal reflection fluorescence microscopy, or TIRF. The idea behind this technique is this. Rather than directing the excitation light straight at your sample, which would show you a field of G-actin monomers, you can direct the light at your sample at an inclined angle. Past a critical angle, there will be a neat phenomenon called total internal reflection at the cover slip water interface. This phenomenon prevents most of the excitation light from entering the sample, but allows an evanescent wave of excitation light to penetrate only 50 to 100 nanometers into the sample. Thus, TIRF microscopy allows you to obtain a very thin optical section, assuming that what you want to look at is at the surface of the cover slip. This means that if you want to observe individual actin filaments growing, you will need to attach them to the surface of the cover slip. So how could you do that? Well, since actin filaments bind to myosin, a good strategy would be to coat your cover slip with myosin. However, you don't want the myosin to move the actin, as it normally would, since that would make the actin hard for you to focus on under the microscope. You can solve this problem by treating myosin with N-ethylmaleimide, or NEM, a chemical that inactivates the myosin motor but still allows myosin to strongly bind actin filaments. Thus, by coating a cover slip with inactivated myosin, you can get the actin filaments that form in solution to stick to the cover slip surface within the 50 to 100 nanometers illuminated by the evanescent wave. The actin monomers diffusing throughout the rest of the chamber, on the other hand, are not illuminated and thus do not fluoresce, greatly reducing your background noise. Okay, so using this technique, you can see how actin, shown here in green, self-assembles. But can you learn about how actin binding proteins mediate actin assembly? To do this, you would want to label not only the actin, but also the actin binding protein, so that you can visualize both simultaneously. So can you now describe how to measure actin assembly through both a bulk measurement technique and a single molecule method? Could you apply the principles of these techniques to other scientific questions in cell biology? Below this video, we have posted several discussion questions. Think about them and post your answers on the discussion forum. Thanks for joining us for this episode of Neat Experiments in Cell Biology.